Good afternoon. Thank you for joining me today for another installment of TDG's Digital Health webinars. My guests today are Drs. Maxime Kennison and Chi-Ming Ho. Dr. Kennison is professor and chair of the Department of Anesthesiology and Perioperative Medicine at UCLA's David Geffen School of Medicine. Dr. Ho is a UCLA Distinguished Research Professor Emeritus. Dr. Ho leads an interdisciplinary research lab to apply artificial intelligence to personalized medicine. Today's webinar will focus on enhancing surgical outcomes through blood pressure monitoring. Professor Kennison and Ho are both doing some exciting research on blood pressure monitoring, which they have agreed to share with us today. We will start with a presentation from Professor Kennison. So Maxime, when you're ready, feel free to share your screen, and looking forward to hearing your talk.
Okay. Good morning, everybody, or good morning still. My name is Maxime Kennison. I'm the chair of the Department of Anesthesiology here at UCLA. It's my pleasure to be here, and thank you for giving me the opportunity to present some of the work we do in our lab, and that's been supported sometime in part by actually TDG and some of the grants available. So what my research group focus is on blood pressure monitoring and hemodynamic monitoring during surgery and in the intensive care unit. Just to give an idea of the size of the significance of the problem, we have in the United States, about 40 million people are undergoing surgery every year. It's about 240 million people undergoing surgery worldwide every year. And in the ICU, we're talking about 5 million patients being admitted to the ICU in the United States only. And that was before the COVID-19 pandemic. So it's a pretty large patient population. The outcome of the surgical patients are, for the most cases, is pretty good, because not every surgery are high-risk surgery. But still, there are a lot of complications following surgery, and this has a cost for our healthcare system, a cost for patients, and that's what we'll discuss together. Blood pressure is one of the key physiological parameters that allow life to exist. We could not live without a low blood pressure. Blood pressure is a key determinant. If it's too high, people die of hypertension long term. If it's too low, people die very quickly. It's an acute hypertension that lasts more than a few minutes, leads to death. We cannot live with a blood pressure that's close to zero or extremely low. But even a period of slightly low blood pressure, and just for people who are not clinician or physician here, the hypotension, which is a low blood pressure, is defined as a blood pressure less than 65 millimeter of mercury for the mean arterial pressure. Even a short period of time, just a few minutes of blood pressure less than 65 millimeter of mercury during surgery has been shown to lead to an increased postoperative complications such as kidney failure or myocardial infarction or even death. And blood pressure is a very finely regulated physiological variable that can go below 65 in a very, very short time without even us paying attention. Over the past 10 years, there's a lot of evidence and a lot of expert consensus basically emphasizing the importance of preventing hypertension, low blood pressure during surgery. It's so important that actually in the anesthesiology community and the American Society of Anesthesiologists, through the Anesthesia Quality Institute, has made prevention of intraoperative hypertension one of the key quality measures for the management of surgical patients. And what it means to be a key quality measures for anesthesiology and surgery, that reimbursement from CMS, Medicare, are going to be linked to some extent to the prevention of intraoperative hypertension during surgery. So it's associated, we know it, to increase postoperative complication in surgical patients. And now it's becoming a quality measure that physician and organization will have to optimize in order to make sure that they deliver the highest level of care to their surgical patient. The question is, how do we measure blood pressure during surgery? So the good news is that blood pressure is measured in 100% of patients undergoing surgery, at least in the United States, in Europe, and in other countries. It's measured in 100% of surgical patients using the device that you see here on these patients, this blood pressure cuff. It's a curve that probably everyone here listening to this webinar has experienced once in their life. So a curve that you put on the arm and that goes up and down and measures the blood pressure. The good thing about this blood pressure curve is that it's non-invasive, just a curve that you put on the arm. The problem with it is that it doesn't provide a continuous beat-to-beat blood pressure measurements. You measure blood pressure every 3 minutes or every 5 minutes. So if you measure every 5 minutes, you have 5 minutes window, which between two measurements, you have no idea what the blood pressure is. If you want a continuous blood pressure measurements, you use an arterial line. That's a very invasive device that you put in an artery that can be on here on the wrist. And that's going to measure through a transducer the blood pressure beat to beat. The good thing about this is that it's a beat to beat measurement. You can detect changes in blood pressure as soon as they happen. The problem with it is that they are highly invasive. There's a lot of complications associated to this. So in the cost-benefit ratio of decision-making for an anesthesiologist or surgeon taking care of these patients, if a patient is undergoing a very high-risk surgery, it's about 10% of the patient population undergoing surgery, it's okay to place an arterial line. If the patient is not undergoing a high-risk surgery, 90% of the surgeries, we use the blood pressure cuff. And the problem is that by measuring blood pressure with the blood pressure cuff, we are missing a lot of events of intraoperative hypotension that leads to postoperative complication that we've discussed before. So that's just to give you the baseline for the conversation about hypotension and the epidemiology, the significance of this. But now let's look back at the past. How did we used to practice surgery and anesthesiology? This is a picture that's a reenactment of the first anesthesia procedure performed on a patient in 1846 at the Massachusetts General Hospital in Boston. And as you can see at the time, 1846, you had 9 physicians to deliver anesthesiology to one patient. Today, when you go to the operating room, you're surrounded by technologies. You have all the monitors in the world, blood pressure, heart rate, oxygenation. You have devices here administering medications, the vasopressor and everything, but everything is disconnected. There's a lot of technologies. And as you can see, there's not 9 anesthesiologists delivering the care. There is one anesthesiologist behind their phone taking the picture here. So the physician to patient ratio has changed consistently, and we are now surrounded by technology. One of the problems is that these technologies do not communicate with each other. A good part of this technology that you can see here on this monitor, we measure heart rate for every patient. We measure the oxygenation, a pulse oximeter waveform for every patient. And we monitor blood pressure intermittently for about 90% of these patients. So this data is already here. And I'll show you how we can use this data. One of the things that we've worked as a lab for many years is to make these technologies work together, to make them communicate. And that leads us to this concept of closed loop system. That's, for example, a case that was done in Belgium, in Europe. That was published in 2017, so already 4 years ago. In this case, what you see that we monitor the patients with the monitor that you have here. We have a monitor here that monitors the blood pressure continuously, the cardiac output. The data from this monitor go to a computer here. This computer is equipped with a software that's called a closed loop hemodynamic software that controls the administration of fluids and vasopressor through this pump here. You have these 2 monitors here. One is for the depth of anesthesia. The other one is for the pain management. These 2 monitors send information to this computer here that is equipped with a software that was developed by some people on our team. And this software is going to control the administration of anesthetics and opioids. In other words, here in this system, the anesthesia was totally automated under the control of computers to lead the administration of vasopressor, fluid, but also anesthetics during surgery. And this is one case report, but in some places in Europe, centers are using the technologies that are presented here that we've developed. And some places are moving toward a more and more automated system. That's the system that we developed, and the IP is owned by the UC Regents. It was developed when I was at UC Irvine. And this has been licensed to Edwards Life Sciences in 2014, and actually has been FDA approved in December 2020. Now, a first step toward a fully automated system for fluid management during surgery. That's the paper that was published just this year. That was the IDE study that led to the FDA approval that was performed by investigators at the Cleveland Clinic. Other ways to control the blood pressure this time is to link the actual blood pressure measurements to a computer that's going to administer drugs automatically to the patient to maintain the blood pressure in very narrow limits. So, for example, again, this is a monitor that measures blood pressure, cardiac output. The information goes here on this computer that controls the pump here with the phenylephrine, which is a vasopressor to maintain the blood pressure within narrow ranges during surgery. And here in this experiment, for example, what we do is that we induce hypotension in some animals. We induce the hypotension with drugs, and we don't do anything. So what you can see here, you have 8 pigs. Each line is 1 pig over about a 4 hours period of time. And you can see when we induce hypotension, the blood pressure goes from 80 to about 40 millimeter of mercury. And in the other group, what we do is that we do the exact same experiment where we decrease the blood pressure, but we also associate our automated system that's going to give vasopressor automatically. And what you can see here is that the system is going to maintain the blood pressure within narrow limits without intervention from a human being, automatically, a little bit like the air conditioner would do, where you measure the temperature in a room and the air conditioner controls the administration of airflow, either hot or cool, and maintains the temperature at a steady limit. What we do the exact same thing, but with a physiological variable, and especially with blood pressure. That's another example of the use of this technology, where you see here blood pressure in a patient during surgery for about 2 hours, where the blood pressure is under the control of a human being. You see the blood pressure goes up and down, doesn't stay within narrow limits of what is recommended. Then you put the patient under the automated system to control the blood pressure, and you see that the blood pressure for another 3 hours remains within the target for blood pressure that is known to be the best target. And when you stop the system and you let the human being do, again, the blood pressure goes up and down. And we know that human beings are not very good at very mundane tasks. We are very good at processing complex information in a very timely manner, but we are not good at repeating the same task again and again and again. And that's what automated system and closed-loop system do together. Now, how can we apply artificial intelligence and not automation, but AI to this kind of paradigm? So one of the systems that we have developed a few years ago is a system that's going to predict when a patient is going to develop hypotension. Because once you have the system that automates the administration of medication, you can react when the blood pressure goes down and the automated system starts giving the drugs. But the best would be to be able to predict before the blood pressure goes down so that you can start administering the medication. So in order to do that, years ago, we created very large databases of high fidelity blood pressure waveforms, ECG waveform, SpO2 waveform. We started doing this back in 2012. And now this slide needs to be updated, but we have more than 5 million minutes of continuous recordings of physiological waveform. And what you can do using artificial intelligence is to identify patterns in these waveforms that are going to be extremely predictive, for example, of hypotension. And so I'm going to go pretty quick on this one. But in other words, what we do, we take the arterial blood pressure, the waveforms, we apply feature extraction techniques to this waveform, then we apply some predictive modeling, and we are able to predict hypotension with a pretty good specificity and sensitivity. The strength of this system is the numbers of features that we are able to extract from each arterial pressure waveform. In other words, for one arterial beat, one heartbeat, we're able to extract up to 2.6 million features for just one arterial beat. And of these 2.6 million features, we're able to detect that when 23 of them happens to be exactly at the same time, they are very predictive of the patient developing hypertension. The system, the same way, is also commercialized by Edwards Life Science for a system that's called the hypertension prediction index. At UCLA, with these databases combining the EHR, the waveforms, and the genetics data, we also have pretty large opportunities to develop this kind of system. There is one system that we've developed with the group at the computational medicine with Dr. Iran Halperin and his team in computational medicine. And the concept is as follows. If you're able to record more than 5 million minutes of high fidelity waveform recording, like you see here, for example, you have arterial pressure waveform, you have the SpO2 waveform, and the EKG waveform in green. If you have multiple millions of heartbeats with these multiple waveforms together, what you can do potentially, theoretically, using AI, is to use a waveform like the EKG waveform and the SpO2 waveform and train a computer to impute what will be the arterial pressure waveform without even measuring it. In other words, if you were to be able to measure the EKG waveform, which we do in 100% of patients undergoing surgery, and the SpO2 waveform, which we do in 100% of patients during surgery, we could impute continuous and non-invasive blood pressure monitoring for 100% of patients using software. The idea is that when you have enough information, you can teach a computer how to impute another waveform without measuring the waveform itself. So that would replace the invasive arterial pressure, but that would most importantly allow to measure blood pressure continuously in all of these patients for which today we just have the cuff. And instead of giving the blood pressure every 5 minutes, you would give it continuously without an invasive device. And that's the system that we've developed recently. You can see here, for example, an example of the true arterial blood pressure in green, and in red is the predicted blood pressure, the imputed blood pressure. And you can see the recording of this data with the true blood pressure in green here, recorded over 1,000 seconds, or 10,000 seconds, sorry, and then the imputed blood pressure using the AI system. And you can see this also in another patient. In other words, what you see here in red is that you measure the blood pressure without measuring it. You're actually imputing it, and you can have it over time in a non-invasive fashion. And again, this technology is owned by the UC Regents through IP that's been developed here. So I'm done with my presentation. That was a high-level overview of what we do, and I hope you learn a lot about this topic, and I look forward to Dr. Ho's presentation.
Maxime, thank you very much for sharing all that information. I had a few questions. I'm curious, you mentioned that there was this closed loop, there were a couple of closed loop systems, the hemodynamic software and the anesthesia and analgesia system. Where are those in terms of getting into the market? Have they been licensed by somebody, or are they up for FDA approval? What's the status of those?
So, there are a few groups in the world working on this topic. There are maybe one group in the United States plus our group. The other group is in Texas. A few one in Canada, 2 in Europe, and 1 in India. So far, our system, we were the first one to have our open loop. So the first step toward fully closed loop system to be FDA approved and CE marked. And to the best of my knowledge, that's the first system to be actually commercially available as an open loop. And it's the FDA is kind of conservative about it because we are basically automating a task that was meant to be done by a physician, even though it's a very low level task, but it's a task that's been historically done by physician. And we've approached it in a stepwise manner with Edwards Life Science to first get the open loop software approved by the FDA, because the software is the software that controls the closed loop system. And this one has been approved by the FDA. And the next step is then to work on the connectics between the monitors and the pumps. And the hope is to have a system that's going to be approved within the next 2 years, that's going to be fully automated for fluid. The vasopressor system that I presented is a system that's also the IP is owned by UC. That's been licensed to a startup company that's called Perceptive Medical. And that's now developing the system plus the pump system together, integrated with the monitor and the closed loop system. And that's a very early stage. So the hope is having a FDA approval within the next 4 to 5 years. But we have the experience of doing it, at least for an open system, open loop for fluid management. So we go like stepwise in terms of the approach for this.
Excellent. That's exciting that one of those systems could be available as early as 2 years from now. You'd answered a follow-on question of mine. I was wondering for the assistant fluid management software why that had taken so long. I think from 2014 to 2020. And so what I heard you say was the FDA is being extra conservative because this is something that is traditionally handled by a physician, and so they're being extra cautious in terms of the steps required to have these approved. Is that a correct assessment?
Yeah, I think it's a correct assessment. It's also the fact that it's been, in the monitoring business, when like companies producing monitors, where they just measure physiological variables, they live in the regulatory environment is very different from intervention or drugs or pharmacology, right? Where you treat, or you have an intervention or a device that treats, that's invasive. And the companies that produce monitors are, for the first time, for example, I think for Edwards Life Science, that's the first time that they had to go through their critical care business for the monitoring business to go through an IDE with the FDA. So that's also a learning experience, I guess, on both sides. And the FDA they don't have a lot of experience yet with approval of automated system that supposedly take the physician outside of the loop, even though our system actually never take the physician outside of the loop because the clinician has full control over what the system does. But still, it's a shift in paradigm compared to what we do today. So I think that's why it's been a little longer.
Yeah, that's fascinating. Well, before I move on, I know that my colleague Marby put something in the chat for our guest. If you have any questions for Maxime or for Qiming, please put them in the Q&A, and then we'll have some time to answer those as we continue with the talk. So Maxime, thank you again. That was really interesting to hear more in detail what you're working on. So now I would like to move to Professor Ho and hear about the research that he is doing in his lab. So Qiming, if you'd like to share your screen.
Okay, do you see my screen?
Yes, looks great.
Okay, so today we're going to talk about the personalized control for hemodynamics in high-risk surgery patient enabled by AI PRS platform. And so Max also mentioned about the importance of this problem. So among all high-risk surgery patients, around about 900,000 patients undergoes cardiac surgery per year in the US. And the post-operative AKI occurs about 5 to 30% of them. And even a very short time. And some report has been say even 1 minute of intracooperative MAP less than 55 or 65 millimeter Hg associated with the AKI. In hospital, mortality of a patient with AKI is around 20 to 25%. So in other words, poor perioperative hemodynamics management is a contributing factor to post-operative AKI, and which is associated with increased mortality. So this is a record of a pressure monitoring during about 50 minutes. So you can see the pressure start with 75, and it happens a very sharp drop to 60 something. Okay. And that's already in the 55, 65 range. And then it come up, and then sudden sharp drop, increase again, and coming down, and then has gradually up and downs, and then suddenly shoot up. Okay, so there's a lot of fluctuations, and which including two modes. One is a very sharp drop or very sharp increase, and one of that is going up and down a lot, but with smooth in time. Okay. So that's one of the things we need to take care. Another thing so how do we take care of these two drugs? One increase the blood pressure, one decreasing blood pressure. And the most time is doing by dose titration. And so basically it's a try and error approach. So that's one of the challenge we're taking. And Max already shows something as very good ways to take about this type of control. Another of that is how do you do it with a very small deviation from an MAP. Okay, so this MAP is, say, let's set at 80, and then how much we can make fluctuation in very, very small range so that help the situation. So challenge of control the hemodynamics. Suppose the pressure is low and we want to bring back to delta P. It's a very simple equation. So that's the things we want to bring back. We already know this value. And we want to know is what is the dose. Okay, so it's related by a sensitivity. But the sensitivity depends on individual patient. Every patient is different. Another thing is that the sensitivity can drop with time, especially when tachyphylaxis happen. And then it will drop very, very much. Sometimes can be 5 times or more less sensitive than at the early stage of the control. So another thing is we are not only adjust one drug, we are trying to adjust 2 drugs. One is going up, one is going down. And the drug-drug interaction becomes serious. So 10 years ago, we started working on these drug-drug interaction problems. And basically, we use that to study the combinatory drug. How do we optimize the combinatory drug? In this case, that was our perhaps the first experiment and did in the in vitro case. So we take 6 drugs. And if each drug has 10 doses, and that's 1 million possibilities. So how a doctor can decide what is the best drug out from this pool and what is the best concentration for each of them. Okay, and so what we do is we study HSV1 infection. Yeah, HSV1 is a herpesvirus. So in a petri dish, we put a HSV1 virus and also the host cell. Then we try to find out what is the best drug dose combination out from this 1 million. So we use very simple artificial neural networks. And what we want to do is we use the training data to train artificial neural networks and what is the drug dose response surface. The output is very surprised. Okay, so the drug dose we put into the body, and in this case, put into the cell, and then we measure the efficacy. In this case, this efficacy is what is infection, how to inhibit the infection. So it come out, it's a very simple surface. Vertical axis is the efficacy, means the percentage of inhibition of the rate. The 2 horizontal axes are the dose. And you can see it's end up with a very simple mathematical function. It's the second order and the polynomial type of function. So that simplifies how to find the optimal drug and optimal dose from a very large pool of possible combinations. And after we get this function, and then we only have to do, say, 3 drugs, we have only to do 6 tests. We can find out the coefficient for each of the patient at that moment. So in this case, we have only 2 drugs. So the equation, the function become very simple. As the first order of these 2 drugs, second order, and the drug-drug interaction. This term is the most important part. It can either increase the efficacy or decrease the efficacy. Depends on the coefficient. And that coefficient, these coefficients depends on the person and it depends on time. Okay. And without this, you have to go back to 1 million possible combinations. So that really helps a lot. So what we do is we see how these 2 drugs interactions from the data which we got from the patient data. So we found out it's not a curved surface. It's a flat surface with a slight slope. So that tells us it's not a nonlinear drug-drug interaction. So we don't have to work on this two drug interaction. We can use this back to work on this equation. And the thing is still the sensitivity, which depends on person and it depends on the time. So we use these AIPRs things we apply to many these models we did in clinical trials. Okay, we did for liver transplant, we did for cancer, and we did for infectious disease. This case, the post liver transplant is very similar to what we're doing now. So tacrolimus used to suppress the rejection of the liver, but the tacrolimus is neurotoxin. So if there's very narrow range, which is a sweet spot for not reject the liver and will not cause neurological side effect. But the range is very narrow, 2 nanogram per ml. This is a control patient. That's doctor control by titration method. So at the first, if the dose and it's a little bit too high, so he want to drop down to the range, drop a little bit, then start drop a lot, then start to increase, but it doesn't increase that fast. So it takes a long time, 10 days to get back to the range. Okay, then there's our patient. So our patient, we remember that function. We take a few calibration tests. After a few calibration tests, and then the fourth day, we can go to very close and can make the tacrolimus level very close inside the range. Okay. Recently, we found out if we control that very well, our patient can discharge 50% shorter time than the control patient. And we tried this method for organ transplant, for cancer, and for infectious disease, all doing pretty well. And especially for cancer, the response rate is 100%. Okay. You know that usually cancer patient to the chemo is 25% response rate. Okay, now let's talk about what we try to use this method to apply to the current case. So we use a rat, and we want to control the rat pressure in a very narrow range around a target value. So the dose we can control is concentration and dose, and we have to watch out how does that vary with function time. So we use 2 drugs. We cannot use fentanyl now because that's a control drug. So it's nitroglycerin, sorry. So that's the first experiment. And what do we want to demonstrate? The 2 drugs which are response almost independently, except with a very small linear dependence. Indeed, when it's too low, we inject drug, and too high, we inject another drug, so we can go up and down. If we adjust the amount of the dose into the rat, and we can gradually reduce these pressure fluctuations. Okay. However, during this test, we don't know what is the sensitivity, but we demonstrate the AIPRS tells us both of the drugs work almost independently. So that's good news. So that was before COVID. So after COVID, then we come back to look into the issue about S. Okay. So the first few experiments, we have to establish the population average sensitivity. So we doing many rat, and we do the experiment and use different concentration, different dose, and etc. And we find out what is the average dose of the sensitivity of the rat. So, okay. Now this is a real experiment. So with 85, then we use a drug simulating a cardiac effect, so drop the pressure. When drop the pressure at this moment, then we inject the drug. How much drug we inject based population average dose. So when we inject that, so it shoot up, okay, too high. And so after going down, and then we injected the other drug, try to reduce the drug, and we also put in the population average the sensitivity. Then this time is better, going down and almost stay within the range. Okay, so obviously the sensitivity is very important, but it's not only sensitivity of the population sensitivity. We have to get the individualized sensitivity, and also has to do with dynamic calibration at this point. So that's what we do, is how do we come out individualized sensitivity for that rat at that moment? What we do is at this moment, we based on the population average sensitivity, inject a little bit of the drug. So it will going up. When going up, during this amount of time, we can calibrate what is, because we know the pressure difference and we know how much drug we put in, then we can calculate the individualized sensitivity. So we inject the individualized sensitivity, and should be possible to bring in to the range pretty nicely. So that's what we use this method, and we will be able to get individualized sensitivity of the rat at that moment. And when time goes by long, and we have to calibrate each time, because the sensitivity change with time dramatically, even for the same rat. So that's experimental. So we integrate all the things together, then that's an experimental result. So you can see the pressure pretty constant with simulating the cardiac effect, and it can drop. Okay, drop to that moment. Then we, based on the population average sensitivity, then we inject the drug. So it rises up, and the state that doesn't never get into that means drug is down. Okay, so at this moment, based on the data, then we calculated the individualized sensitivity, and we can keep that within very, very right in the range. So, sorry, what happened? So let's recap the situation. So it goes down, and then we inject the drug based on population average sensitivity. It rises up and it reach almost a plateau. The plateau means the drug is finished. Okay. So based on this slope, we know the individualized sensitivity. And this time we inject, we know the sensitivity, we know pressure difference we want to bring in. We inject the drug, and which can go in there and stay constant. Okay. So now let's do another case. If the blood pressure is up, and when it's up, then we, based on population average sensitivity, we give a calibration shot. And at this moment, we will be able to know what is the individualized sensitivity. So we put the right dose in there, and it should stay within the range. So that's experimental results. You can see the pressure in the range and the rise. When rise to that point, and they keep dropping, and at this point, we try to inject the drug, try to reduce the pressure. So inject that, and it does reduce, but it doesn't reach it does not reach the boundary. It reach in the red zone value, and then it starts again. We put a drug according to individualized sensitivity. We can bring into that and stay within the range. Okay, so these two experimental results basically we have full confidence we can accomplish what we want to do, because at a large pressure difference, and we demonstrate already we can use personalized sensitivity and bring that into range. So rest of that is see how precise we can control and how narrow we can control that range. Okay. So the next problem is we have to do a lot of experiment to find out the time response and of the system, et cetera, and then we can control that within very narrow range. So the prospective preclinical test, we need to do the next step. We want to establish a minimal level of delta P prime, and also as we have to be able to demonstrate it is a dynamic response, a dynamic calibration, and a continuous update of the individualized sensitivity. And then we want to see how difference of each rat different from the average population average sensitivity, because all that will be important for the design programs. Then we will start to do retrospective clinical trial. So we'll collect the patients with the most difficult patient to control. They have kidney, liver, or heart problems. Okay, and we want to collect those things, and we check out this approach we're doing, because these type of things will give you a sudden change of the pressure drop, and a sudden change is very hard to predict from the back history. Okay, it's very much like a computer. It's like a stock market, and there's a lot of software will be able to predict when the stock market regularly fluctuate up and down. But when there's a sudden drop, then that will be hard. And that's a sudden drop, and this time we lose the money. That's also a sudden drop will really damage the organs of the patient. So from the retrospective, we want to go through all this very difficult case, and we determine the personalized sensitivity, and then we dynamically try to update and see whether we can get it done. Another thing is very important. We have to improve the safeguard of the software. Okay, and because the next step we're going to go to prospective. Go to prospective means we base on the population average sensitivity, and in the retrospective clinical data, and we dynamically update the personalized sensitivity, and we see how fast we can bring into the range, and how small we can keep that within small level. Again, the safety guard of the software always need to be to be updated all the time. So it takes 6 months for us to finish the prospective preclinical test, and we are start to apply the retrospective clinical trial, the IRB. Then after we get the retrospective IRB, so at this moment, we get a lot of data from the prospective and the retrospective, then we can start to apply for the prospective clinical trial. And then it will depend on how many we can do the clinical trial, and how much time we can enroll the patient. So this is not done by myself alone. It's a very 7 or 8 people are involved in this, and also fund is supported by the UCLA Innovative Fund. So that's the end of my talk.
Qiming, thank you very much.
You're quite welcome.
So I had a quick question. I know that we've just filed a patent for some of this work. I wasn't clear on the slides. Where were you in the next steps? I couldn't tell which, as you went down the slides or that progress, where you were today and what was going to happen over the next 6 months.
Okay, so which I showed the 2 videos, right? You see the pressure change, okay? And that's the stage we are, okay? And but also you can see the videos which we can sharply bring it back to the range, but we still it's not sharp enough, because we have to make sure the drug almost reach a plateau, then we inject the other one. But if we inject before the drug is finished, first drug is finished, then we inject the second drug, then we have to go back to look into the drug-drug interaction. Okay, Then we see how do we improve that. Okay, that's one thing. And another thing is we doesn't know how the system responds yet. So how fast we can really bring back to range, how fast we can lift that back to the safe blood pressure. Okay, so all that we tested in a prospective preclinical test. Okay, then after that's the beginning of the zero. That's you see the green bar, which is 0 to 6 months. So we are at the 0 point. Means we prove it will be done. It can be done. Okay, and then now we have to test from preclinical to a lot of tests, and to get how fast we can do it and how accurately we control in a small range. Then we will do from the retrospective clinical trial to test on the real data, especially for the ones which has very hard to control, for the one which has a lot of sharp changes. Okay, after that,
got it.
I have to apply for the prospective.
Right.
We prove that we can handle the very sharp drop or very sharp increase. Okay, and bring into the desired MAP range. And also I'm sure we can get the delta P prime into a very small range, much smaller than what we can do now. But I cannot answer that question until the test has been done. Okay.
Yeah, that will require more data. And we actually have a question from the audience relating to that from Leo Wang. And the question is, how much training data does the AI model require for the dose optimizations you mentioned at the beginning? And there's a follow-up question from Leo, which is, and does your group use conventional manual approaches to obtain the efficacy data?
Okay, so first thing is in the first, at the beginning, we talked about AI PRS method. Okay, maybe I can go back to the share. It'll be easier to talk. Okay, for this one, we do the training. Okay, this training is try to find out what is the drug dose response surface. That we use 500 sets of training data from the in vitro experiment. Okay, so after we have done and we discover this function, and that is a function enable all our personalized medicine. Because after we get this function, for example, 2 drugs, we only need to do 6 tests to solve this 6 coefficient. That's all. Okay. So in other words, we can reduce a large number of training data you need in the ordinary AI method. Reduce from millions or 10 millions. Sometimes we did a 10 to the 12th search space. We only need to do few, maybe 10th of the test, and we can solve the function, and we know exactly what is our efficacy at that moment for that person. That's most important. Okay, and if we cannot get the efficacy for that patient at that moment, we cannot optimize the dose, and the patient may not have a good response at all. Okay, so in that case, 2 drugs, we need a 6 test. Actually, we have a trick, we can even reduce to half. The thing is, in the personalized medicine, we cannot do big data AI. You can, if sick patient, you can say, I want to do it on your body to get 100 tests. Okay, so but with this discovery of this function, we can do a few of the tests, and we can get the best one for that patient. And not only the best drug combination for that patient, also with continuously updating in time. So let me see, dynamically we adjust the dose all the time. That's how we can keep it a constant. Okay. So that's the power of that function. So what is the second question?
Let's see. So that covered the data, and then they said, does your group use conventional manual approach to obtain the efficacy data?
For this experiment, no. At this test, yes. Okay. This test, yes. And for this test, we integrate the blood pressure readers, catheters, and syringe pump and the computer into automated system because a human cannot respond that fast. Especially, we have to continuously update the individualized sensitivity. Okay, you can see the equation is very simple. Okay, but still you have to read that and calculate, and that's the problem. We have to do an integrated system, but it's a very simple calculation in here.
Yeah, I love that approach, and I love that there's this. You're doing different things. So Qiming, the thing that's interesting here on the slide is that you have this average drug dosing for the population, and then you do these predictions for the individual drug dosing, which you very quickly modulate until you're able to get a really good model for that person. So that's very interesting. And then what I heard Maxime doing is they're able to get, what I would call synthetic continuous monitoring by using the cuff measurements, which are either every 3 minutes or every 5 minutes, with this massive data set. And that allows you to do this continuous monitoring of blood pressure. So both of these are seem like useful approaches to me to medicine that is personalized to the patient. So just for time, it looks like we have about 6 minutes left. I want to remind the audience, if anyone has questions, they please put them in the Q&A. I had one question for Maxime, and that's so I understand now where Qiming's research is going. What are the next steps for you? What are you focused on in your lab at this point, Maxime?
Well, we have a few focus. The first one is that we keep on working on the prediction of hemodynamic instability. That's one, right? And so everything converges together. I think with the blood pressure imputation, what it gives us as a next opportunity is to apply all of the different intelligence systems that we've developed before, but to also use them, but use the imputed waveform as the input instead of using invasive waveforms. So can you predict hemodynamic instability using the non-invasive system that we've developed? Another part of the research is to also learn. This part is not really dedicated to technology development. It's pure physiological research. It's how can you better understand the physiology by applying computer science? Because, for example, if you're able to predict hypotension, the prediction is based on the statistics, but the statistics does not really reflect any physiological truth that we know of. So the reverse engineering of this system could help us better understand the physiology. And then there is a part of the research that we are developing that's more the infrastructure of the research, is to link the EHR data, the intervention, the drugs, the waveforms data, and also to add the genomic data with the Atlas project. So that's an infrastructure that we are developing. And so every time you do the research, it's like you have like development on like project and specific goals with scientific objectives, but at the same time you develop infrastructure for the next level of scientific goals that you have. So that's how we process the two parts together.
Great. And for those physiological models that you're developing, do you already have partners at the university, or are there particular skill sets that you're looking for in the research community to assist you with that?
So the main expertise that we work a lot with computational medicine. I also have partnership with other universities, with Carnegie Mellon University, Pittsburgh, UC Irvine, UCSF. So we already have a group like this, but the group that computational medicine, we work a lot with them. We work with Mihaela van der Schaar, who's in engineering. But the expertise that we need clinical expertise to label all the waveforms to understand clinically what we are doing. We need computational medicine, computation science expertise to do the analytics, and then also some physiological expertise, physiology and especially cardiovascular physiology expertise.
Great. I know that a good portion of our audience is from UCLA and the research community. So
there is also a part of the expertise, if people are listening to that we always, it's like the people who are able to develop graphic user interface. So it's a graphic display of the information. So that also we have an expertise with it. We also work with people in simulation, because the first step for testing this system is in simulation. And people with expertise on the human-machine interface. I would say that's the level of expertise we're looking for.
All good things to have for your project, for sure. So just for time, because we're 2 minutes to the hour, we're gonna wrap up. So first and foremost, I wanted to thank my guests. So Qiming and Maxime, thank you for sharing your time and all the work that you're doing. What I never got into is I actually know a few people that have had some less than ideal outcomes after surgery from issues like the ones that you've covered. So this is near and dear to my heart, unfortunately. And I'm looking forward to this getting out there so that all surgeries can be more successful and not run into issues associated with blood pressure management. And then for everyone that's watching, if you want to go back and review this, today's webinar will be available on TDG's YouTube channel. And of course, all of the work that we've discussed that was developed at UCLA is available for licensing. And so if you would like to learn more about how to license these inventions, you can email TDG at info@tdg.ucla.edu. So thank you all for your time. And I look forward to having more professors in the future talk about digital health for this audience. So thank you all and have a great day.
Thank you very much. Thank you.
Yeah.
Bye-bye.